Hello, and welcome to the Boffin Buzz, the podcast for anyone fascinated by farming's future. Well, hello everyone, and welcome again to another edition of the Boffin Buzz. And I'm really, really excited today because, as many of you know, there's nothing I enjoy more than talking to scientists, and especially scientists who are exploring the wheat genome. Well, I'm really, really excited to introduce today Cristobal Uauy, Professor Cristobal Uauy of John Innes Centre. So good day, Cristobal. How are you?
Doing great, Tom. Nice to be here.
Fantastic. Well, it's great to have you here, Cristobal. And also, I should say, congratulations on being nominated as Farmers Weekly Game Changer of the Year. So I saw that that was in the Farmers Weekly recently, and I believe you're going to be there at the Farmers Weekly Awards in October. So fingers crossed to see whether you actually get the award. Did you know that you were being nominated?
No, I was quite surprised. I was actually quite surprised, Tom, when I first heard, and it's a real honor in a way to be considered for that, because at the end of the day, that's what we're here for. We want to actually affect change in farming.
Yeah, well, absolutely. Well, that's what I hope we're going to explore today. Now, I've been to John Innes Centre on several times, and I have to say, every time I go and I talk to you, Cristobal, I go slack-jawed in amazement at the things that I see there, and I've said this to you many times before. You know, I can't wait to get these things out into the field. But let's talk about some of the genetics that you've been looking at, and let's also talk about our plans to bring these out into the field, because this is really, really exciting. And we're just on the cusp of what I think is a really, really exciting sort of step change in where we're going with genetics. But first of all, could I just ask you to sort of give a brief overview of the work that you're actually doing at John Innes Centre?
Yeah, so I lead a team that's working on wheat genetics and genomics, which is basically studying the DNA of wheat and trying to find how we associate sequence or the genome with particular traits, so that we know which genes control particular traits. And with that knowledge, then we can find better versions of those genes, or we can help the breeders to select for those genes in their varieties. For example, like midge resistance and things like that, where they can select with a molecular marker, like a COVID test for wheat. They can check if it has it or not, the midge gene, and then they can recommend that into their varieties.
Yes, that's right. And now you've been working specifically with sort of techniques to change the genes. I think so. Could you just sort of give us a little bit of an insight into the technologies that you've been using?
Yeah, I mean, in terms of, I'll put it in context perhaps, just to make sure we
Yeah,
kind
sure.
of know where the technology fits. So if we think about the variation that we see in our recommended list, that all comes from natural variation. And these are mutations or changes that happen naturally in the wheat genome.
Yeah.
And these mutations are quite common. So every seed that you harvest from the field will have 100 mutations with respect to the mother plant. So it already has 100 mutations, but it's relatively small numbers within the big and very large wheat genome. So we
So
can use that.
how do these mutations happen? I mean, we all know about genetic variation that we see in the
Yeah.
field. You know, why a Skyfall is different from a Zayat and a Bamford or whatever. But how does that actually happen in the field then? What?
They happen naturally. So when the plant is growing and it's dividing, and it's, the cells are dividing, sometimes mistakes happen with UV light, for example. That's why we have to put sun cream, so our DNA is not mutated. In the case of plants, they're in the sun, they're getting mutations, and basically when the plant is making more copies of itself or the cells, sometimes a mistake happens, and often that mistake is repaired, but sometimes it's repaired incorrectly. And when that happens, you have a little change in DNA. One letter is different, and then that generates something new. It could be good, it could be bad, but it generates that novel variation. And these are the 100 mutations that every seed of wheat will have with respect to the mother plant.
And the thing about wheat, as you've told me in the past, Cristobal, is it's quite a complex genome.
Yeah.
It makes our genome as humans look really quite simple. Could you expand on that?
Yeah, so the wheat genome is about 3 to 4 times larger than our genome, actually. So it's a very big beast. And the example I say is that if we were to print out the wheat genome on an A4 piece of paper on both sides and stack it up, each individual cell would have the equivalent of a pile of of notes of sheets higher than the shard. So it'd be 320 meters tall, just one cell. And our job is to try to find which one of those pages contains some information that might be of use to farmers. And of course, the cool thing now is that because we have new technologies to sequence genomes, just like we sequenced the human genome 23 years ago, we've sequenced now the wheat genome 5 years ago, we can now read all those letters, and the searching makes it a little bit more, let's say, simpler to do that study that we can now do.
Right, but it must be still like sort of needle in a haystack
It's
territory.
completely one page in the shards haystack kind of thing. Yeah, so it is. And those 100 mutations that are happening will just happen randomly across that whole genome. And sometimes they'll change one page that gives an instruction for something that's useful, or sometimes they'll change something that might make ons appear. So these off types that we see in the field usually are just changes that are happening that make the plant look slightly different, a little bit taller, shorter, flower early, flower later. And breeders and humans have been very good at selecting those changes for something that's useful to them. But because we only have 100 per generation, it's relatively slow. So the way that we've also can do it is doing what's called mutagenesis breeding, or tilling, which is this approach. And in
Right.
this case, what we do is that we take a bag of seed and we purposely mutagenize it with chemical agents that mutagenize it, or we subject them to UV radiation directly. So these are just like we know that there's some chemicals that mutate our DNA. The same thing will happen with DNA of plants. So we take the seeds, and instead of having 100 mutations in every seed, we have 500,000
Right.
mutations randomly across the genome of wheat.
Right.
And
So
this
so
will
what you're
generate new
doing,
variation.
yeah, so what you're doing there is you're taking that variation that happens naturally, from UV light and so on. But what you're doing is you're subjecting the seeds to sort of intense stress, if you like, through chemicals and UV light and things like that, in order to and radiation and things, in order to generate masses of mutations. Is that the sort of thing?
Exactly, exactly. So what we're
Now,
doing is
now
that we're
this is
changing.
this is technology that's been around for a while, isn't it? What we're talking about
Yeah,
here,
it's been 90, 100 years we've been using radiation and chemicals. So
right?
many of the varieties that we grow, even barley like Golden Promise, is a variety that was generated through mutagenesis breeding of these random mutations
Okay,
and then
right.
finding one that was better. And many of our fruits and vegetables come from that because the breeding cycle, when you don't have a seed, takes very long. So in the case of vegetables or trees, there's a lot of this type of ways of generating more variation than happens naturally, just because the cycles are so long to go from, let's say, from a seed to actually a fruit. So they want to accelerate that cycle. So
And
it's something
and
that's been
this
happening and that we've eaten for the last 90 years.
and this. So you talked about this is called mutagenesis,
Yes,
um, and
mutagenesis.
and then as I understand it, there are different types of mutagenesis as well. So and I know that you've been working with tilling. So is that the technology we're talking about here?
So the technology of tilling is also with mutagenesis, but what we do there is that it's like organizing a library. So we take these individual seeds, and because we can now sequence their genome, we can then sequence them, and we'll have these stacks of DNA information, like the shard for every one of the different plants. So we'll have a library, let's say, of books, 2,000 or 3,000 of them. We sequence their genome, and therefore we know where these 500,000 mutations are, which are randomly spread, but we've organized them. So that means that instead of having to go and find a mutation in those 3,000 plants, I can just go online, and within a few minutes, I can say, if I want to mutate or change this gene, I have these 30 or 40 different versions of the gene, and I can then go to my seed store, get the seeds, and I will have that in the seed that I have selected. So it makes an organized way of seeing all these mutations that are random, but it's much more organized. And that's what
I
tilling
see.
in essence is.
I see. And so this is technology that you've been using for some time in order to make new varieties. I guess what's new about that is that because the wheat genome has now been sequenced, and I think you said that was 5 years ago, it makes your job a hell of a lot easier. It's like having rather than having to go into a library and look through all of the books to find the one that you want. It's all sort of computer referenced, and you just say, "I'm looking for this," and it says it's over there.
It's exactly what it is. So before we had to open each book one at a time, and it would take us maybe a month to find for one particular gene. Now it takes us minutes, literally seconds, to find that because we've organized it all and we have it in public databases with colleagues at EBI in south of Cambridge, so we can then find those mutations straight away. And that was work that took a long time to do, but now that it's done, it's now public there, and we've used it, and breeders have also used it to find new versions of the genes that they think are important for their particular traits. And in the
Right.
case of wheat, the really cool thing is that wheat has this very large genome because also it's what we call a polyploid. So it has 3 copies of every gene. And by having 3 copies, it means that it's like, you have the same thing written 3 times. So if you want to change it, you cannot just change one. Often you need to change the 3 copies to have a visual effect on the plant, so you can actually see something different.
Right.
So natural variation allows us to go so far because it changes usually 1 copy at a time. But if we want to change 3 copies at a time, it will be very hard to find that. We can do it by genetics, by crossing, but that takes a long, long time. And in
Yeah.
the case of tilling, what we can do is that we can find those variants, those variations in the books, get the seeds, and then do the crosses in a very purposely manner and in the same background of, in this case, of cadenza, which is a UK wheat.
I see. What so you can look through your library. You can find the cadenza that's got the genetic variation in each of the three genomes, and then
Exactly.
you can cross it so that you've got kind of like a triple cross. If you
Exactly.
yeah,
And when we do
I,
that, suddenly we're seeing variation that we haven't seen before. And that's a
right,
really cool thing, that this variation is there, but it's kind of hidden because unless you combine it, you don't see it. It's like the lights. If I turn off half the light switches, there will still be plenty of light. You wouldn't notice that much difference. But as soon as I turn both light switches off or the 3 light switches off, then suddenly it's dark and you see the
right.
you see the difference. And wheat has that complexity that it has these 2 or 3 switches for every gene, and therefore, unless you take 2 or 3 at the same time, often the changes are very subtle. So now we've been able to see a whole new range of variation, and this is in terms of starch types and starch qualities by colleagues here, or in disease resistance that before we just didn't see because the plant was basically has one mutated, but you didn't have the 2 or 3 genes mutated at the same time.
I see. Well, we'll look at some of those specific traits in a minute, but before we do, everyone's talking about CRISPR at the moment and gene editing,
Yes.
and I know that you've been working in this. So, can you just tell us how does CRISPR differ from the tilling that we've been
Yeah,
talking
so
about?
we had these 100 natural mutations that I said at the beginning, Tom, and then we have these 500,000 random mutations that we can do with the mutagenesis or tilling, and they're organized, but they're still randomly across the genome. We don't control that. And in the case of CRISPR, what it is, it's called CRISPR-Cas, or it has 2 elements. It has a search box and it has a molecular scissor. And the way it works is that we can introduce 20 particular letters, and in a combination that gives us a particular word, and that word is usually found only in the genome in one part of the genome. Because now we have the whole sequence, we have the whole book, we can put these 20 letters in a row, and that gives us a unique combination. And what it does is that it takes a molecular scissor to that particular place and it makes a cut. And when the DNA is cut, what will happen is that the plant will repair itself. But just like naturally happens, sometimes it makes a mistake when it tries to repair itself. And therefore, we introduce a mutation or we make a cut, and the plant will generate a mutation into a particular place of the genome that we've targeted. And therefore, that mutation means that we'll have 101 mutations in that seed because it has the 100 that it always has, plus that one extra mutation that's targeted to a particular place. And importantly, at the DNA level, if you give it that seed to anyone, anyone would say that plant has 101 mutations, but they're indistinguishable. The mutations that occur through genome editing or CRISPR are indistinguishable from those that occur naturally, and because it's using a natural mechanism to do it, and hence, that's part of the excitement of the technology, that it's using some of the tools that are out there already in nature, but it's using it to target a mutation in a particular place and study
Well,
it.
Can I just ask you there, Cristobal, because you talk about introducing these sort of line of letters and a very precise cut and so on, and you say that this is a natural process. I mean, I can see that the cut, when it's cut, when the DNA is cut and then it joins up again, and that's a mutation that could happen naturally. But the actual process of doing that, is that a natural process?
So it requires a step, which is what we call this transgenic step. It requires a step where the plant needs to come into the lab, and we need to introduce the machinery. We need to introduce the search box and the scissor into the
Yeah.
plant. Um, but the thing is that once that search box and scissor, that machinery does the job in terms of making that edit, making that mutation in the particular place, in the next generation, we can just identify plants that have the mutation, but that don't have the machinery. They don't have it because the
Ah,
machinery did the job already. The
right.
machinery is not what actually gives the trait. We're not introducing a gene that makes a particular trait like you would in transgenics. Here we introduce the machinery to make a cut. That cut then gets fixed and repaired, sometimes incorrectly, into an edit. And then in the next generation, we can find the plants that have the edit but don't have the machinery. And hence that plant would be, again, indistinguishable from any other plant in the field. It would just carry one more mutation than you would normally find.
I see. Right. So, although the result is something that could happen naturally, we have sort of um, made that result happen through this machinery, as you describe it,
Correct. We,
but
we,
in a
we have to
very
introduce,
precise way.
it is very precise, but we do have this step that we do need to bring it to the lab to introduce the machinery based on
Yeah.
the current technologies we have. And that was why the Nobel Prize was won by Jennifer Doudna and Emmanuel Charpentier. These two scientists won the prize because they understood the mechanism that was happening in nature that others had discovered, but then they
Right.
adapted it into a simple way that we could do it. And we're using this technology in humans, in animals, in plants, and so on. And it's a universal mechanism, but you do need to introduce the machinery to make it happen as we currently have it. And then in the next generation, when the plants grow again, that machinery will, just by simple or traditional genetics, it will just move away, and we select the plants that have the mutation but don't have the machinery. So that plant will be again 101 mutations with respect to the other plant that has 100 mutations next to it.
I see. Right. But now I want to move on to the specific genes that you've been working on
Mhm.
in your lab, and they're known as the MADSbox transcription factors, I believe. Could you just explain to me what it is?
So this match box. So there's a family of genes that are, let's say, are master regulators that control other genes. So not all genes do everything. So some genes do some biological processes in terms of uh, making a particular enzyme a certain way, but these genes control other genes. They're kind of the boss
Right.
around, and they're the boss in many traits have to do with development of flowers and development of the structures that will then form the grains. So these matchbox genes tend to be involved in anything to do with the development of the flower. And
Right.
what we found is that we found a variation in one of these genes that was in a wild weed called Triticum polonicum. And it has, if you look at the spike, it has extremely long blooms and lemmas. So it looks very awkward with respect to a typical wheat spike. And it has these massive grains, very long grains. So we started studying this with colleagues here, James Simmons and Nikolai Adamski, 10 years ago. And we
Right.
wanted to understand which was the gene that was controlling these very long blooms and very long grains. And
Okay.
by having the genome, we were able then to do all the genetics and find that it was one of these Matchbox genes that was controlling this trait.
I see. Right. So, what you're talking about here is that we're not talking about the actual gene that makes the grain size, so to speak, but it's a gene that says once the grain gets to a certain size, I'm going to the gene acts to stop it growing any bigger. But in this what was it, Triticum polonicum,
Polonicum.
that gene isn't working. Is that the way it
Well,
works?
so in this case, what we think is happening is that because the flower is bigger,
Yeah.
the cavity into which the grain grows is larger. So you have a bigger, let's say, a bigger area that the grain can grow into. So the
Right.
grain will not be encapsulated by these two structures that we typically see in the grain, that when we thresh a grain, we just open them very easily and the grain is inside. Those are much larger now. And that cavity allows the grain to grow into a bigger area, and it can just keep on growing. There's no pressure to keep it a certain size. So we think that the gene is acting in terms of affecting the size of the flower organs. So, and then it also is working inside the grain to make it keep on growing for longer. So there's this dual effect that we're seeing.
I see. And as I understand it, this, Triticum polonicum has been looked at by botanists for years and years and years. But it's only recently that you've discovered the gene that's responsible for this. Is that right?
That's right. So Linnaeus, a famous botanist, classified as a different species originally as Triticum polonicum. And then we
Yeah.
found that this one mutation, this one gene, and mutation this one gene is actually what triggers it. So I can take a
Wow.
modern UK wheat and make it look like a polonicum with this one mutation.
Okay. So that's the big burning question, Cristobal. Have you managed to achieve that?
So halfway there, let's say. We have done it, but critically, this Triticum polonicum has this one very important gene that controls this Matchbox gene. And there are other elements as well. So when you make the mutation in wheat and we change this one gene, we get, let's say, we get halfway to polonicum, not all the way. But that's one of the questions we have is like, if we make grains out a little bit longer, a little bit longer, a little bit longer, what's the best, let's say, what are the possibilities that we can achieve? And then how does that actually look in the field? So we don't get a spike that looks completely wild, but we get something that's much longer in terms of the flower structures. And then you get this longer grain. And importantly, in discussions with millers, they tell us that it also has better test weight. So we can actually put 5% more on the back of a lorry than we can with a traditional grain that's a little bit more round. So it also it gives us that benefit.
So now I think you've made this edit in Fielder. Is that right?
That's correct. So because the as we talked before, CRISPR requires us to introduce the machinery
Yeah.
to make the edit. Initially, to introduce the machinery, let's say again, about 3 or 4 years ago, we could only do it in a few types of wheat. Uh, and Fielder was one of them. And Fielders have a variety from the 1970s from the US that no one cares about. But in the lab was one of the few lines that allowed us to make those edits. So at this initial phase, we did it with that. But we can maybe talk about it later. But the really exciting thing now is that that machinery can now be introduced into recommended list varieties. And that's,
Right.
again, these technologies don't usually happen in once. You have the genome editing, the CRISPR technology, but really I think the game changer for me is that together with having the genome and then also having the ability to introduce these edits into recommended list varieties directly rather than having to take cadenza or something from the 70s and cross it back into the 19 or the 2025 variety, because that will basically lead us to a 1990 variety and we start to do all the breeding again. So
Right.
we can just come back to that point later, maybe.
Yeah. So let's just focus on this Fielder that you've done. So you've already done
Yeah,
the work in Fielder. You've made the edit. You've got a plant now that has got these bigger, bolder grains.
hmm
And what you've done is you've multiplied up enough of it to put out into a field plot. Is that right?
that's where we currently are. So we have checked that there's no machinery left in these plants. So these
Yeah.
plants are just edited plants, and that's correct. So we bulked it up, and now the idea will be then to pass them on and bulk them even further to go into the first kind of trials in a field outside of our experimental station here at the John Innes.
And that is the really exciting thing about this. So, as some of our listeners may be aware, we've just started a new project at Boffin called PROBITY. So that's a platform to rate organisms bred for improved traits and yields. It's a 3-year DEFRA-funded project, £2.2 million project, and we've got several industry partners, including John Innes Centre, I'm delighted to say, and also Rothamsted Research, as well as other partners involved in this. And we're taking the first 3 gene-edited plants from the greenhouse, and we're bringing them out into the field. And Boffin members will get the opportunity to grow these in, I think it's 2026 is when we'll have enough
Yeah.
multiplied up to grow these in the field. And so there are 3 of these gene edited. They're the first 3 gene edited plants to come out into the field. And there's been a specific change in the legislation that has allowed this. So this is the genetic hang on, I'm going to get my it's the genetic engineering
Precision breeding.
precision breeding genetic engineer.
Yeah, genetic
Which way
technologies.
around is it?
It's the Precision Breeding Genetics Technologies, I think, Act.
That's it, Genetic Technologies Act that was passed last year, 2023, and that allowed England is the only place in Europe now where we can grow gene-edited crops in commercial fields, and that is the opportunity that we're taking advantage of with this project. Now, just to emphasise here, they are not commercial varieties. These are not varieties that will come into all commercial fields and will be marketed as such. This is very much a research project. The reason we're doing it is to look at the specific traits. And the other thing to note about these traits is that they are in a non-commercial background. So as Cristobal says, we're looking at fielder in this particular instance. But this fielder is one of the varieties that we are bringing out into the trait. Cristobal is currently multiplying it up. I think we're shortly going to be harvesting, or could even. I don't know whether the harvest has actually taken place yet,
Not
Cristobal,
yet, but soon. Yeah.
Absolutely, and then bring it out into the field, as I say. There's going to be another year of multiplication that will take place as part of the platform, and then we'll be bringing them out onto the field to about 10 commercial growers. We're also going to be working with Dyson Farming, who are going to be Dyson Farming Research, who are at the cutting edge of research in this sort of field, and they're going to be looking at the agronomy of these varieties, because that is the thing that I think is really exciting about this, Cristobal, is that you've been talking about the genetics of getting these bigger, bolder grains, but we all know that wheat compensates. That a bigger grain will then compensate in less tillers or things like that. And so it's not until we get them out into the field and we begin to understand the agronomy behind this that we can actually really use this new trait to our best ability and explore, how we can treat it, how we could be completely rewriting the agronomy rulebook here, and that's the exciting thing from a research point of view that I think we're going to be exploring here.
Yeah, I think for us, Tom, is exactly that. That we can try them. We first try them in a pot, then we go to a small scale trial, 1 meter square trial, then 6 meter square trials. But we have one set of agronomy that we use. We can maybe do two, but really the conditions that we use on site, we try to make them as close as we can to what farmers are doing. But of course, it's very different when you do it at a larger scale, and then when you start actually implementing agronomy trials. So I think that for us is really exciting. And initially, as we said, it's going with fielder, but then in the future we want to bring in additional varieties as well that are closer to more competitive varieties. But at the end, that's what we need to do, and also different soil types and so many other things, variables that we currently have very limited scale in terms of what we do, and we need to see them out in the field.
Yeah, and we're talking specifically here about so DEFRA has made a new term for the crops we're going to be looking at here, which is precision bred organisms, or PBOs. So
Correct.
could you just explain what the difference is between, what makes a PBO a PBO?
So a PBO would be a precision bred organism. So it's let's say any organism, in this case a plant, that would have gone through a genome editing approach. These molecular scissors, a cut, and a repair mechanism that introduces a change. And importantly, the PBO means that there is no foreign DNA. There's no extra machinery left behind, which is the machinery that we put to make the cut. So the machinery has to be out. So it would be a plant that will have a few changes. Maybe one letter is different, or maybe has a small deletion of maybe 10 or 15 letters. And that changes, what we hope is that changes how that particular gene behaves and how that plant behaves in the long run.
Yeah, and I guess the important thing there is that if it's a genuine PBO, then it's absolutely fine to come out into the field because it hasn't got any of that machinery that left in the plant.
Correct. So that primary legislation was passed last year, and then DEFRA passed secondary legislation in terms of how they hope to see things in the field, and that's why we can actually do these trials now, because we can release them to the environment. As I said, we can grow them in the field. They don't pose any threat according to scientific evidence and to DEFRA, because again, these mutations, if you imagine 100 mutations that happen naturally in all the hectares of wheat that we have in the UK, the whole genome is mutated. It's already there. It's just very hard to choose it from, right? So we're going to grow these plants. We can grow the plants out, but now the secondary legislation from the Food and Standard Agency, FSA, is what we're missing to be able then to go into the food chain. So currently we can grow them, but they cannot enter the food chain as of today. But that secondary legislation is currently in Parliament, and we'll see how the new Parliament proceeds with that.
Yeah, and that's one of the interesting things about this project, Cristobal, is that we're going to be growing up quite large amounts of this wheat potentially in 2026. At the moment, as the legislation stands, we don't know what we can actually do with this wheat because the legislation hasn't yet been written to say whether it can be fed to cattle or fed to humans or how it can be used. That's as I see it. Is that right?
That's where we currently are, and FSA have put forward a very much science-based structure of how these should be regulated, so that there's a transparency about which PBOs will come to market. So it'll be a public registry they've proposed, which is excellent, and then they'll be tested or they'll be evaluated a certain way that is science-based and it's risk-based. So it's actually not about the technology, but about the risk and so on. And that gives us security in terms of consumers and citizens. But importantly, it doesn't put a huge onus of regulations that means that the only people who can bring these to market would be the big multinationals.
Yeah.
Institutions like John Innes or collaborations between breeders and farmers, or even farmers and John Innes, can now start thinking about breeding this because the technology is relatively simple to implement. And that means that if the legislation is science-based and risk-based, that then allows us to make sure that then if we're doing things like small changes like we're thinking about, that can actually get to market relatively quickly and without the onus of having to go through trials that would imply millions and millions of pounds, which would actually only jeopardize and would basically make it only available for multinationals. So that's something that I think is also important, that legislation is science-based and is risk-based to make sure that technology can get to farmers as soon as possible if it's sound science.
Yeah, no, let's look again at the actual specific fielder that we're going to be bringing forward here. So, as I say, what we're hoping to explore in the field is the agronomy around it, and the how we're going to be applying nitrogen, how we're going to grow it, what sort of tillage practices, and so on, really get the best out of this trait. But what do we actually know about this trait so far? I mean, we've talked about the bigger grain size, but what does that actually mean in terms of, have you done much in the way of tests on it to see what specific weight, for example, it is, what sort of protein levels, is this likely to be a super milling variety, or is it going to be a feed variety? Have you any idea of where it's going to fit?
So the tests that we've done have been looking at protein and also test weight. So
Right.
protein-wise, although we get a bigger grain, we don't get dilution, which was really important. So that was one of the
Okay.
first surprises we had because we expected having a bigger grain would dilute the protein and the percentage of protein would go down. But we actually maintained the protein, which is really important. And
Right.
then the test weight is what I was mentioning before. I think is quite interesting because in the trials that we have done with our current agronomy, we see the bigger grain, but we don't see an increase. We do not see an increase in yield, but we see this increase in test weight, where we actually get 5% improved test weight, and that's across the board. So all the trials we've done, small plots, large plots, every time we've done it, we've seen this. And that's actually very interesting to millers, of course, because that's one less
Mm.
truck out of 20 on the road. If we can
Sorry,
actually,
just to define, when you say test weight, is that the same as specific weight?
yeah, specific weight. It's the
Right,
same. It's the
specific.
same terminal. Yeah, or hectoliter
That's
weight. Yeah. So
that's
we can
really
pack
interesting then.
5% more in the same volume. We can pack 5% more in the same volume, and we think it has
Yeah.
to do with the shape of the grain that is slightly more elongated, and it just packs in a slightly different way than the more traditional round grain. And importantly, the flour yield that we've seen is the same one, about 79%, 0.79, 0.78 as a typical variety. So That's why we want to really get enough volume to be able to go through milling trials and see how that will behave. Again,
Yeah.
it's still in fielder, it's not the best variety, but then right behind it, we can then bring it in other more elite varieties for our group ones.
Yeah, and that's the other aspect of this because, as you alluded to earlier, it's not just about the fielder here. That you've also developed the technology for that you can apply on other varieties than the recommended list. Is that right?
Yeah, so that's one of the really exciting developments is that until very recently, we could only transform field or bob white. These varieties are not very relevant to UK or international farming at the moment. But over the last 3 or 4 years, there's been developments led by uh, colleagues around the world, and we are using a technology developed in UC Davis in California that was also implemented here at John Innes with Sadi Haita. And what they were able to show is that they could now use this technology to improve the efficiency and overcome the recalcitrant of some of these UK cultivars to transformation, to put the machinery in. So we have now successfully been able to transform Skyfall and several other recommended list varieties and also many international varieties. And that means that we can then put the machinery into Skyfall, make the edit, and then the next generation find a Skyfall with 101 mutations, but without that machinery. So that's really exciting because we can now put it directly into UK varieties and national list varieties perhaps in the future. And importantly, perhaps that will not be the variety straight away, but when breeders start crossing, they start introducing this new variation into the gene pool of UK varieties. And that's important because if they have to cross with fielder, they say, well, that's going to dilute all our efforts of the last 50 years. But if they cross Skyfall, that's a parent that they're currently using. So they can now take a Skyfall with an improvement and bring it into the gene pool, knowing they'll have all the benefits of Skyfall plus the new traits that we're putting in.
So, have you made that edit yet?
It's happening. So we made
Right.
edits with other colleagues. So we're using several edits. So there are traits related to yellow rust, which is work that's been led by Diane Saunders here at the John Innes,
Right.
works on different starch compositions. And then we're also trying to increase the grain number of wheat by manipulating or changing a particular gene. And these are all edits that are now, instead of doing them in Fielder, we're now putting them in Skyfall. And that's,
Uh-huh.
again, we're using Skyfall because that's a variety that we can get, we can transform very easily and it works really well. It's milling, so it gives us kind of a nice balance. But theoretically, this can be done on any UK variety now. And
Right.
of course, Fielder will be okay, but then if we can get into Skyfall and show those effects, the take up by breeders and hopefully by farmers that can grow Skyfall, they've seen Skyfall, they know how it looks, and now with an improved yellow rust, for example, or with different starch compositions, or with improved grain number and grain size, those will be really fascinating things that we can start looking at.
I see. So you could take a variety. I mean, the Skyfall's a bit long in the tooth now, isn't it? But you could take a variety like Bamford, for example, and make the same edit. Is there any reason, if you can do it in Skyfall, is there any reason why you can't do it in Bamford?
No, so far what we found is that most of the varieties, not to say all, but to now, the varieties that we've tested from the UK recommended list, we can transform. The efficiency might
Right.
be different. So when you do 100, you might get 70 or 50 or 40, but you can. And before that was zero. You couldn't get it. Now we can. It's just a little bit more work perhaps with some of them, but we can now get the machinery in, make the edit, and then the next generation get out the machinery and just leave that edit inside, leave the edit in the DNA. Yeah.
I see. Now, I think you've also been looking. You mentioned some of the other work that some of your colleagues are doing, like Diane Saunders and so on. I think you've also been looking at other specific genes. So the one that we were talking about that's in the fielder is that's had the VRT A2 gene knocked out. Have I got my terminology right there?
The VRT-A2 gene is correct. We haven't knocked
That's
it
right.
out. What we've done is
Oh, okay.
actually changed the changed the way it's expressed. So
Uh-huh.
uh, this is one of these matchbox genes that controls other genes, but everyone has a master. So this gene also is controlled by other genes. And basically, what happens here is that there's sequences around the gene that tell it when to turn on and off. And in Polonicum, in this wild wheat, there's a section of that that's changed, that's different, and that means that the gene is turned on slightly higher in a different place than it would be in a
Uh-huh.
traditional wheat. And what we've done is that we've generated an equivalent edit that the one in the wild into a fielder. And the question of why don't we do that by just traditional crossing is because it takes us 15 years to get anything that would look remotely like fielder, trying to do it in that way. So we've now made the edit in fielder, and now we're going to test how that's going to look. So we get the longer grains and so on, but now we'll see how that behaves when we put in the field in terms of seeing yield, test weights, and so on.
So I guess that begs the question: if you've done it with one gene, are there other genes that you're looking at specifically as well?
Yeah, so as I mentioned, we're also looking at, we're working with one gene that is this molecular break on the grain size. So there's a gene that we have used with the tilling approach, with this mutagenesis approach, that we can definitely get grains that are 30% larger or 25% larger. They have
Wow.
more protein, but they don't have more yield. So the plant
Right.
compensates. Okay.
Okay.
So, and it compensates by having slightly less grain number. So what we're trying to do is that we're now adjusting a gene that, again, there's a natural variation in this gene that basically allows you to, instead of having maybe 3 or 4 grains per every spikelet in a spike, it has 1 extra grain. So what you do is you increase the grain number. So
Uh
we now
huh.
want to combine the 2 traits into a variety with this genome editing approach to see how, when you combine the 2 traits together, how they look. Again, traditionally, you could do it by crossing perhaps the mutant, but again, it would take a long time. And often what happens is that the mutations are in different backgrounds. So you need to combine them, and then you have lots of other things happening. So here we will have lines that are basically genetically identical, apart from one having mutations in the genes that make the grain bigger, another in the genes that make more grains, and then we'll have the two together, and we'll see
I
how those plants
see.
behave. And of course, there's where we're going to need working with Buffin and other farmers to actually
Yeah.
see how those behave on trials on farms with different agronomy.
And that's a really exciting thing about this, Christopher. I mean, I think the fielder that you're currently bringing forward, that we're, as I say, hopefully going to have in field trials in 2026, is exciting in itself. And when I say field trials in 2026, I hope we're gonna have some material that we're going to be bringing into small plot trials next year, as early as next year, which is really exciting. But that's just the start of where we're at, and as you highlight there, with this technology, there are going to be different genes. We're going to be stacking genes and so on, or edits, should I say, in order to explore exactly what we can do here, and then bring those out into the field and see how they express themselves in the field. I mean, as I see it, we've got something of a pipeline of varieties coming through here, haven't we?
I think what we have is that, let's say for relatively simple traits, I put simple between quotes,
Yeah.
grain number, grain size, starch composition. For some of the traits that we know the genes, we know which of the 320 meter pages, which page is actually controlling the trait. We can start doing the edits. But let's say for more complex traits, maybe if we're really manipulating yield, we don't know what will happen if we change the grain
Yeah.
number and the grain size. Maybe the plant will make less tillers, or maybe the plant will do something else. But importantly, these have always been correlations, like, oh yes, this variety has more tillers and has smaller grains. They've always been that type of study. And I think importantly, we can now ask the question, okay, if I make 20% larger grains, what does a plant do? And that makes less of them. Okay. Now, if we make it make more grain, how is the plant going to compensate? What happens if we grow it under conditions that have more CO2 maybe in trials, and see if we
Mm.
feed it more energy? So those are things that we can now do that before it was very correlation. Oh, there's, someone told me that this variety has this. Now we can actually do those tests. And importantly, that's how I think we're going to be able to start understanding some of these more complex traits, understanding the elements, combining them, and then we're still a little bit away of being able to do predictive breeding, of saying, oh, if I change this, this, and that, I will have higher yield in this location. But I
Yeah.
think that's where we hope to get to. But we still are at the phase where we're doing individual genes, maybe 2 or 3 genes, putting them together, seeing how they work. But in the future, you can imagine where, just like we have a library that I can now find mutations in every gene of wheat and go into the library, very soon we probably will have technologies where we can then take any variety, take Bamford, as you said, and actually generate mutations in the different genes and combinations that we can now test. So perhaps breeding will look quite different, because instead of just relying on natural variation, we can now say, okay, we take that, but what happens if we change these 30 or 40 genes, which we know are important, we make changes in them, and then we breed for the best combination of those that perhaps we don't know yet, but when you breed for them, you'll select the best one. And then we can study, what did a breeder select in the UK, actually by changing these 10 genes, they found the best combination of yield in the UK. And maybe if they went to Devon, another part, they would find another combination of 10 genes that gave the best
Yeah.
yield there. So that's, I think, where we're going to go. So currently, one or two genes at a time, but in the future, probably a bag of seed that has changes in 10 or 15 important genes, and we breed on top of that. And I think that's a very different way of breeding, and that's hopefully where we get to. And that empirical selection by breeders will then inform us back to say, this was selected, and we need to understand why was that selected, and then that's going to be a cycle. So that's why connecting with the farmers and the breeders for us is so important, because that information will feed back to us rather than just kind of from the lab to the breeder to the farmer.
Yeah.
It'll be more a circle, I think, a virtuous circle.
And that's exactly what I hope we're going to be delivering through Probity, and why I'm really, really excited about this project and the fact that we've got farmers working directly with you, Cristobal. But I want to move on now, because there's another exciting gene that you've been a genetic change that you've described to me that you're working on as well, and this is to do with the rooting ability of wheat. So could you just give some background there?
So this was work that was originally done in barley, and these were colleagues in Nottingham and in Italy and Germany, and they were working on barley, and they found that changes in a particular gene, they were actually working on two different genes, changed the angle in which the roots grow in barley. So instead of the roots being quite superficial, they found that by changing this gene, they changed the angle in which the roots grew, and the roots grew more steeply. They grew down more. So they explored more of the surface, or more of the profile going down into the soil. So what we did is that in wheat, of course, because you have these two genes, but you have three copies of each one, we had to use the tilling population, the cadenza mutants that we had. We were able to find the corresponding gene in wheat, find the mutations, and combine them. And when we combined
Right.
them for gene number one or gene number two, when we combined them, we found that the roots became much steeper. And we now have a plant that has, let's say, the six copies of the gene. They have everything together, and we get plants that have cadenza plants that have very, very steep roots, not superficial, but very steep. And again, importantly, instead of comparing lines that have different root types that are very different, we have cadenza with its normal roots, cadenza with slightly steeper roots, and cadenza with very steep roots. And those are now being
Ah,
tested within the Delivering Sustainable Wheat program with colleagues at Rothamsted and at Nottingham. And we're trying to test them what happens, how do they grow under a conventional or a no-till situation? How do they grow in sandy? How do they grow with less water? And to see
okay.
how these plants will behave under these different conditions.
So, what stage are these plants at? So, you've got some material available, have you?
Yes, we do. So we're bulking seed for you, Tom. So we're now currently we have we're going to put next year a big tram line so we can bulk the material, and these will be they're not PBOs. These are just tilling mutants in cadenza, and we have the three combinations together. So we have the control plants and the deep root plants.
Right.
And those are ready. So we're bulking the seed now to hopefully be able to share it with Boffin Network next, not 2025, but 2020 or harvest 2025. We hope to be able to share them with seed to have enough kilos to actually do proper trials.
So that's for planting in 2020, autumn 25. It's
Correct.
cadenza, I think you said, wasn't it? So that
It's
would be autumn
cadenza.
2025, wouldn't it? Well, yeah.
Correct.
So and that's another thing that we're particularly excited about. As you may know, we've got the Truth Project that is currently going on. This is
Yes.
a 3-year, again, DEFRA-funded project with 5 industry partners, where we're looking at the interaction between roots and soil. And we have root rangers, our farmers at the moment, who are doing intensive soil sampling on wheats in their field. Now, this year, they've just been looking at their own standard wheats and doing some work, particularly looking at the rhizosphere, but also looking at the rooting structure within their crops. And looking at so we've been doing DNA analysis of their soils, then rhizosphere, and then just bare soil. Part of what we're doing is bringing to field a new sensor called the PES sensor,
Hmm.
which it smells the soil basically, and should give you a quick in the field analysis of the microbes that are in your soil. Now, next year, we're going to be looking at, this autumn, we're going to be looking at one or two interesting wheats. Those root rangers are going to be trialing some interesting wheats. And then the year after, is when we've got some Watkins lines that your colleague Maria Hernandez Soriano has been
Yes.
looking at. So these are Watkins lines that have biological nitrification inhibition. So this is the ability to control their own nitrogen uptake, which no other commercial wheat has got. It's just been identified in these Watkins lines. So we're bulking those up at the moment. They'll be available in 2026 harvest season, along with a couple of durum wheats with really interesting rooting characteristics. And we're hoping to bring your wheats, Cristobal, in with that program in that year is the plan. So watch this space if this is something that interests you. We are currently recruiting for root rangers. We've got 10 root rangers at the moment, and we're looking to increase that number to 20 for the coming season. So if you're interested, then please do go to the Boffin page, follow the link to the Truth Project, and apply to become a root ranger. And then you'll be involved in the trial. You'll be the first to hear about these exciting new varieties that we're bringing through. But look,
and just to add one thing, Tom, to that, just to
yeah,
say, so these are the mutants in cadenza, which are done with this mutagenesis approach, this tilling approach. But importantly, if there's any benefit of those traits, if we see that there's any merit, then immediately we can then go and recreate those mutations with CRISPR, with genome editing, into any elite variety and transfer that trait directly like that. So I think that that's the critical thing, is that before we would have to take cadenza and then cross it to the elite variety of today. And instead of saying that, I say, well, actually, let's get an elite variety of today, a national list line. Let's get those edits in, and then the breeders can start crossing with that rather than crossing with technology or, let's say, genetics of 1990. So I think that's where these technologies are not, kind of exclusive of each other. They complement each other. And in this case, this technology, tilling, was the best to do this initial trials. But now for deployment, we could do with tilling, but then that would just mean that there would be a huge risk and financial burden for the breeders to work 30 years to get it into something that's approachable to UK farms, or we could get it into UK varieties. And of course, most likely will need to be crossed, but that would mean maybe 5 or 10 years rather than the 20 or 30 years that would require now. So I think that's where the technologies help each other to at least start accelerating that movement into varieties and into your fields.
Yeah, and that's quite an important point, isn't it? Because tilling, as I understand it, so going back to what you were saying right at the beginning about tilling and the many, many mutations it makes, as I understand it, the tilling lines that have a particular trait in them also tend to carry a yield drag or some other disability with them. Is that correct?
Well, they carry 101 mutations, the 100 that always happens, the one that you want, and another 500,000 that you don't want. So
Yeah.
that's the problem, that they carry stably 500,000 mutations in their genome, and we know that because we sequenced them, right? So that means that then breeders have to take not just cadenza, but cadenza that has one beneficial trait and possibly 500,000 mutations that could give it linkage drag. But again, it's very hard to know if it will affect yield because we will need to grow proper plots. But let's say in general, some of them will not be positive. So then you need to weed them out. You need to get them out of the system. And that's one way
Exactly.
of doing it. And
Years
that
of
would just take
crossbreeding.
lots of crossing, lots of
Yeah.
breeding. Or you can say, well, I'll put those 3 mutations that I know make the very steep roots into Bamford or into Champion or whatever, and just put it directly, or Diamond, for example, and then go like that. That could be the way. I think
And
that's where
and, and sorry, just to clarify there, Christobel, when you say I'll put that into Bamford, you're talking about CRISPR
with
there,
CRISPR, exactly.
yeah, exactly.
Exactly, because then we don't have to put the baggage of 500,000 mutations. We say we take Bamford or we take Diamond, we make the edit, and that plant then will have the edit. Again, we do need that step of putting the machinery in, but in the next generation, 5 months later, we take it out and we just leave the edits into that plant. And I think that's the exciting part for us, that we can move these discoveries that often are we find these very interesting traits. We think they could have potential benefit, but then if someone needs to invest 15, 20 years on them, it's a lot to ask the breeders to invest that time. So instead of saying that, we say, actually, let's put in something that's much closer to market.
Yeah.
And then they have the risk, they can de-risk that strategy, and they can really say, okay, let's give this a go, because they don't need to waste, or let's say spend 15, 20 years into something that perhaps might not behave perfectly well in the future. They can then do these trials straight away.
No, that sounds great. Well, I mean, Cristobal, we could talk all day about gene editing, and
For sure.
nothing would thrill me more. But we must bring this to a close. But before we do, there is the, if you like, the moral question that we want to answer here. I mean, what we've been talking about throughout this episode is the scientific discovery, if you like, and that's really what we're looking to do with Probity is bring some of the science that you've been looking at in the lab, bringing that out into the field and saying, okay, what does it do? Does it do what the same thing that you've seen in the lab? How does it express itself in the field? How does it express itself in different scenarios, in a real world environment, in a commercial environment? How does it interact with the agronomy and things like that? But there is the question of, should we be doing this? So as a scientist, Cristobal, you're concerned with, can we do this? And what can we do? And what are the limitations, and so on. And one thing that struck me, so I read recently about an edit that's been made that apparently makes wheat roots much more able to grow, so they can bust through a compaction pan, for example. And it sounds at first sight as though this is a fantastic idea, but then you think to yourself, well, actually, hang on a second here. What we're talking about is a genetic variation that gets round bad farming. Let's face
Yeah.
it. So we shouldn't be asking the wheat to get through compaction. We should be solving the compaction. And this is I just sort of raised that as a thing. I'm sure there are lots of listeners out there who think to themselves, this is all very well, but should we be doing this? Are we just not repeating the mistakes of the past, if you like? What are your views on this, Cristobal?
I think that most of the traits, I mean, that we're working on is trying to go to a much more sustainable angle, to more regenerative agriculture, so less chemical inputs. The BNI trait that you mentioned just recently is trying to reduce the amount of fertilizer that we need to apply to soils. The roots that we're looking at, we're not necessarily thinking about that. We're thinking about being able to take advantage and make a better profile of soil going down. So I think that most of the traits that we're interested in are traits that have to do with more sustainable regenerative farming practices. And I think that that's where most of the scientific community is. Importantly, we're trying to reduce chemical usage and to have the technology not in coming from the outside into the farm in that way. We're trying to hopefully have the technology in the seed so that the plant can defend itself from pathogens and pests rather than have to rely on chemical sprays. We're trying to make the plant be able to better communicate with, as you say, with the soil, to be able to take more nitrogen directly rather than having to put more and more nitrogen into the soil for it to respond. So all of these traits, I think, can be used in a context of agronomy for regenerative farming, for our net zero targets, and so on. But it's not the technology per se. I think it's how we use it. Importantly, with the current legislation we have, it really lowers the barriers for, let's say, smaller companies or these networks that we have between scientists and farmers to really have farmers drive the agenda rather than have the technology drive the agenda and say, this technology is going to save farming, you need to use it, and you need to put more chemicals, bigger tractors, and so on. Perhaps we need, that's why we're having this conversation, to have more farming input and saying, actually, these are the technologies, or this the way we want to use the technology for more regenerative farming, for net zero targets that we have. And importantly, then, instead of having to, rely on a breeding cycle that often limits what genetics can get to the field, we can accelerate that cycle by really making some of those changes in things that are currently grown, and we can actually test them in the field. So I think it's not about the technology per se, it's how we use it, but we need to have the dialogue of the food chain, not just, let's say, the companies that are trying to sell more chemicals and so on, but really have their voice, but also have the voice of retailer citizens, and importantly farmers, to say what they want this technology, how they want to use it, and what they're worried about. And talking to them, they're worried about, will this include make the seeds more expensive? Who controls the technology? How can we deploy it? And those are all questions that we need to have. But importantly, if we don't have it, we're going to fall in the same scenario that we have now of just concentration of a few companies that sell chemicals, sell seeds, sell everything, and perhaps there's a different way. And this technology hopefully can offer a dialogue to allow us to explore those different ways. And I don't have
Yeah.
the answer to that, but I think that these conversations is what we need to make sure technology works for farmers and not just for particular companies that have a business model that's okay. But I think for the targets we have, for the challenge we have as a society, we probably want to have that conversation with you now. And this technology, for the first time, allows to get us genetics to farmers more quickly, which is something that's been desperately missing in our system. And now we have that, and that's why we want to work with you all to do that.
No, well, that's absolutely why I'm looking forward to working with you, Cristobal, over the next 3 years on this project, and hopefully well into the future as well.
Yes.
And it is, as you say, it's about exploring what's possible. It's about bringing it out into the field and answering some of those questions that are currently unanswered. Really, and we really want to involve everyone in that conversation, and that definitely includes farmers, and also, as you mentioned, food manufacturers and consumers. We all need to chip in on this conversation, which we're going to be having with the PROBITY platform. It's going to be a very exciting project, I think, and really earth-breaking. But look, Cristobal, that's all we've got time for at the moment. I mean, I hope you'll come back to the Boffin Buzz over the course of the project and give us some updates as we go through.
Yeah,
I'm
I'd be
certainly,
delighted to, for sure.
yeah. Well, I tell you what, I can't wait to see your fielder out in the field and see how it performs in plot trials and then in our commercial trials as well. And of course, all the other traits that you've been talking about as well as they come through. But look, thank you so much for joining us today, Cristobal. And thank you, all of you, for joining us on the Boffin Buzz. And so until next time, goodbye.